Hey, so all welcome back to my YouTube channel. Today we will going to discuss about a very simple topic but yet very important, that is glycolysis. It is also called as EMP pathway with the name involving their scientist name, that is Embedden Meyerhoff pathway. Okay. First of all, we should remember that before beginning with glycolysis, let me make some concepts simpler for you. First of all, what happens? Suppose this is a cell. This is outside and this is inside part. Okay. Now the glucose from outside wants to enter inside the cell. Okay. It can enter with the help of receptor here, that will be GLUT receptors. Okay. Without the GLUT receptors, glucose cannot enter from outside to that of the inside. So basically different types of GLUT receptors are present. First of all, let me mention you, like in our skeletal muscles we have GLUT4 receptors. Okay. In our brain we have GLUT1 and GLUT3, while in the RBC we have only GLUT1. So you should remember these receptors. Okay. So this is how glucose from outside the cell can enter inside the cell with the help of GLUT receptors. Okay. But what happens in the case of diabetes? Diabetes basically is hyperglycemia. What happens that insulin here is resistant and the GLUT cannot be produced. When the GLUT cannot be produced, the glucose cannot enter the cell and is lying freely, so it can lead to the hyperglycemia. Okay. So you should remember the various type of GLUTs present in the region. Okay. So let's begin with our glycolysis. I will make some simpler flowchart for all of you. First of all, what happens? Firstly, glucose is converted to glucose 6-phosphate with the help of enzyme, that is hexokinase or glucokinase. Okay. Then glucose 6-phosphate is converted to fructose 6-phosphate with the help of enzyme phosphohexoisomerase. Let me mention here phosphohexoisomerase. Okay. Then fructose 6-phosphate is converted to fructose 1,6 bisphosphate with the help of enzyme phosphofructokinase. Then the phosphofructokinase will divide it into 2 with the help of an enzyme, that is aldolase. Okay. It will form DHAP and glyceraldehyde 3-phosphate. The full form of DHAP is dihydroxyacetone phosphate. Okay. So this DHAP and the glyceraldehyde 3-phosphate, they have the interconversion with the help of phosphotryptophan. Then this glyceraldehyde 3-phosphate will convert into 1,3 bisphosphoglycerate. Okay. Then 1,3-bisphosphoglycerate will convert into 3-phosphoglycerate. Okay, 3-phosphoglycerate will convert into 2-phosphoglycerate. 2-phosphoglycerate will convert into phosphoenol pyruvate. Phosphoenol pyruvate will convert into pyruvate. Now let me mention you about the various enzymes. Glyceraldehyde 3-phosphate will convert into 1,3-phosphoglycerate with the help of enzyme, that is glyceraldehyde 3-phosphate dehydrogenase. Then 1,3-bisphosphoglycerate will convert to 3-phosphoglycerate with the help of phosphoglycerate kinase. Then 3-phosphoglycerate will convert into 2-phosphoglycerate with the help of phosphoglycerate mutase. Okay. Then 2-phosphoglycerate will convert into phosphoenol pyruvate with the help of enzyme enolase. Then phosphoenol pyruvate will convert into pyruvate with the help of enzyme pyruvate kinase. Okay, so let's begin with it again. What happens? First of all, glucose will convert into glucose 6-phosphate with the help of enzyme either hexokinase or glucokinase. Then glucose 6-phosphate will convert into fructose 6-phosphate with the help of enzyme, that is phosphohexoisomerase. This PHI is phosphohexoisomerase. Let me write down here. Phosphohexoisomerase. Then fructose 6-phosphate will convert into fructose 1,6-bisphosphate with the help of enzyme phosphofructokinase. Phosphofructokinase. Then fructose 1,6-bisphosphate will divide into two parts, that is DHAP, that is dihydroxyacetone phosphate, and glyceraldehyde 3-phosphate. Okay. Then there will be the isoconversion between the two with the help of enzyme, that is phosphotrioisomerase. Phosphotrioisomerase. Then glyceraldehyde 3-phosphate will convert into 1,3-bisphosphoglycerate with the help of enzyme glyceraldehyde 3-phosphate dehydrogenase. 1,3-bisphosphoglycerate will convert into 3-phosphoglycerate with the help of enzyme phosphoglycerate kinase. This is phosphoglycerate kinase. 3-phosphoglycerate will convert into phosphoglycerate mutase. Then 3-phosphoglycerate will convert into 2-phosphoglutarate, and 2-phosphoglutarate will convert into phosphoenol pyruvate with the help of enzyme enolase, and phosphoenol pyruvate will convert into an enzyme that is called as the pyruvate with the help of enzyme pyruvate kinase. So this is in short about the glycolysis. Firstly, remember the steps, then it will be easy for you to remember the enzymes. First of all, glucose is converted to glucose 6-phosphate. Glucose 6-phosphate is converted to fructose 6-phosphate. Fructose 6-phosphate is converted to fructose 1,6-bisphosphate. Then it divides into the two parts, DHAP and glyceraldehyde 3-phosphate. Then it divides into 1,3-bisphosphoglycerate, then 3-phosphoglycerate, 2-phosphoglycerate, then phosphoenol pyruvate, and then converts into pyruvate, and then learns about the enzymes. Okay, so here I want to mention some important things in this cycle. First of all, during the conversion of glucose to glucose 6-phosphate, here 1 ATP is consumed. Okay, and from conversion of fructose 6-phosphate to fructose 1,6-bisphosphate, here also 1 ATP is consumed. Okay, so basically the 2 ATPs are consumed. Okay, now during the conversion of glyceraldehyde 3-phosphate to 1,3-phosphoglycerate, there will be the production of 5 ATP molecules. Okay, and then from conversion of 1,3-bisphosphoglycerate to 3-phosphoglycerate, there will be the production of 2 ATP molecules. And then from phosphoenol pyruvate to that of the pyruvate, there will be another 2 ATP molecules produced. So basically from glucose to that of the glucose 6 phosphate, 1 ATP is consumed, and from fructose 6 phosphate to that of the fructose 1,6 bisphosphate, 1 ATP is consumed. What are the steps where energy are produced? Energy are produced basically in the step where glyceraldehyde is converted, 3-phosphate is converted to that of the 1,3-bisphosphoglycerate. There 5 ATPs are produced, and 1,3-bisphosphoglycerate is converted to 3-phosphoglycerate. There also 2 ATPs are produced. Further, when phosphoenol pyruvate is converted to pyruvate, then also 2 ATPs are produced. So in total, how much ATPs are produced? So it will be 5 ATP plus 2 ATP plus 2. So basically in total there are 9 ATP. Sorry. Okay. And how many ATPs are consumed? Basically there were 2 ATPs consumed in the above mentioned steps. Okay. So we have to, so aerobically, in the case of glycolysis there are 7 ATP, and anaerobically, in the absence of oxygen there are only 2 ATP. That you should remember. How come the 7 came? As production and consumption. Production is like oxygen is there, and ATP consumption is like oxygen is absent. So in the presence, basically it is 9 minus 2 is 7 ATP. Okay. So this is the amount of this energy consumption. And further I want to tell you about what are the committed steps. So basically during the conversion of fructose 1,6 bisphosphate into 2, this enzyme aldose is the committed step. This is the committed step that you should remember. Okay. And then there will be the 2 substrate level phosphorylation. First of all, in the case of phosphoglycerate kinase, there will be, in this step, there will be first substrate level of phosphorylation. Let me mention over here. First substrate level phosphorylation. Okay. Second substrate level phosphorylation will occur when there will be conversion of phosphoenol pyruvate to that of the pyruvate. Second, it will be here. This is second substrate level phosphorylation. Substrate level phosphorylation. Okay. So you should remember which enzyme is involved in committed step, that is aldolase, and which 2 enzymes are involved in the substrate level phosphorylation. First of all, it is phosphoglycerate, that is kinase, and then it is the pyruvate kinase. And all of you should remember that this enzyme enolase is blocked with the help of fluoride. Okay. And what is the key glycolytic enzyme? Key enzyme for the glycolysis is basically our phosphofructokinase. Phosphofructokinase is the key glycolytic enzyme. And there is something that is called as pasture effect. It is also associated with glycolysis. Basically, pasture effect is inhibition of the cycle of glycolysis. Inhibition of glycolysis cycle by oxygen. Okay. As there, a phosphofructokinase enzyme will be inhibited. So this was all about glycolysis that all of you should remember. Learn the various steps and learn the various enzymes. Okay. So this was all about glycolysis. If you will try to mug it, it will be difficult. You should spell the terms and the enzymes again and again, so that you get very confident with the term. Otherwise it will turn either something, somewhere it will be isomerase and somewhere it will be kinase. So just learn and revise it again so that it fits into your mind. So just thank you for watching my video. Please do like, share and subscribe to my channel. Thank you for watching.